Good morning, everyone. Thank you for joining us today for the RFP grant for 2023. So for today, we're gonna start doing some administration just to make sure that when you are in the Zoom webinar and if you have any question you would like to ask, address your question in the Q&A box, which you can find next to the chat box. You can use the chat box to say hi to us and to say hi to other researchers in the group. But make sure if you have a specific question that you want us to answer after the presentation, make sure it included in the Q&A box. So for today, I'm going to do an introduction of our research grant for today and the funding priority A. And then I will pass on to my colleagues, Dr. Manasa for funding priority B and Dr. Dean Powell for funding priority C. I will conclude the funding priority D as well as how to apply for this grant. And we will have a Q&A session afterwards. So for those who might not be familiar with the Good Food Institute, we are the nonprofit organization that dedicate to accelerate the ecosystem of alternative proteins. We work in three programmatic pillars, which are science, technology, corporate engagement, as well as policy. We also located in 6 different regions globally, and that's the US, Brazil, Europe, Israel, India, and our team here in Asia Pacific. So we are working to enhance alternative proteins because we believe that this is a solution for us to fighting climate change. And our corporate engagement team will build relationship with the world's largest food manufacturer, companies, restaurant, also startup as well as investor. Our policy team will work closely with the government to unlock any regulation or infrastructure needed to bring these products to markets. And in science and technology, the group here with you today, our team of PhD scientists is working to advance the open source to help with the foundational science of alternative proteins. And as a result, create a thriving research ecosystem. And that's why we are welcoming you to the webinar for the grant today. We play a unique role in because we are here to lift up the stakeholder in alternative protein space. So the GFI research grant is one of those ways that we try to promote a more open access research funding into the alternative protein areas. And our aim is to make alternative protein delicious, affordable, and accessible. And the alternative protein within our scope, including cultivated meat, plant-based product, and fermentation, offer exciting research opportunities and tremendous positive impact can come out for the climate and for the global health. So through the GFI research grant program, we have dedicated funding for a specific topic, and we also want to build community, as well as we want this grant program to be a seed funding for you to help unlock any further funding. So far, our program have awarded $17 million in research at more than 100 research topics across 17 countries. So for this year, the 2023 research grant program, we have $3.75 million across 4 topic areas. And this grant is closed for application on September 21st, 2023. The 4 topic areas that will be addressed with this grant is, the first one is Extrusion 2.0, enhancing the traditional extrusion through processing innovation and mechanistic evaluations of protein texturization. The second topic is developing tools and knowledge to promote stemness and proliferation in seafood cell cultures. The topic 3 is data collection and curation to inform the development of genome-scale metabolic model for optimization of feedstock formulation and feed conversion. Last but not least is to improve feedstock availability for food fermentation in biomass and precision fermentation. The award will be up for 24 months for $250,000. There is a potential to unlock additional funding of $100,000 for the project project that involve collaborators with the researcher who are new to the alternative protein field, because we want to bring in new talent and new perspective into the field, and therefore we have this $100,000 options. So let's now move on to the first topic of interest. For the funding priority A, we are looking at extrusion. So as many of us know, even until today, extrusion is still the most common technique used in plant-based meat production, and therefore in alternative protein, because plant-based meat are still the most available alternative protein in the market thus far. However, the extrusion itself still have its limitation, with the current output only up to 500 kilogram per hour, and at the current capacity of produced protein extruded with the size of around 100 micrometer fibers. And this is a lot bigger than the animal fibers. In the extrusion itself, if you were to look at the illustration on your left, the screw section used to control the change in protein ingredient properties, and the die section on the far left is used to control the shaping and solidification of the product. So we want to improve the extrusion by either improve its throughput capacity, as well as provide innovative strategy to replicate the texture, visual, and appearance of conventional animal even further. So for the field catalyst priority A, extrusion, we are looking at the challenge that we are facing with the technology right now. The first one is being the extrusion as a top-down texturization method. We have limitation of how much texturization capability can happen within the food matrix. So it's also difficult to elucidate how proteins interact within themselves and other ingredients under various temperature, moisture, mechanical shear condition. So we have seen some effort of researchers are creating software coupling with unique dye processing, optimizing dye geometries, and also applying these rotating dyes to help enhance the structure of extruded of plant proteins. So the first part of this topic is actually try to see how can we understand better to create a better plant-based meat product that can closer mimic those animal fiber. So as you can see, this innovative dye design or any other design into the extrusion machine itself that might help to deliver a better texture of the protein, as well as the research to understand what's going on within the extrusion. You might be using any sensor or any detector to help understand what happened when the temperature changes, how the moisture feed changes, as well as how the shear force change affect the final products. The other side of challenge for extrusion is its scale. According to the GFI study launched recently, as well as the one launched previously last year, in the anticipating 2030 production requirements, and we have just launched the manufacturing capacity and pathway for expansion, we need to capture 6% of meat market. It will be required, and that's 27 billion capex investment into building an extrusion facility. And that is a lot of money investment to be earned. So therefore, we need to understand how we can actually scale up these production better beyond what we already have now. Right now, the study do not take advance in extrusion into account. So we want to see the synergy of any innovation that you can use on the extrusion machine, and how would that innovation both improve the texture and improve the scalability of the technology. So we have seen some current effort from company like Plextro to have released a high capacity die that can produce up to 1.2 ton per hour. But then again, we need more knowledge in the open access research, and therefore we are focused on these topics. So for the proposal, we will be looking for a proposal that answers the several questions that we have, like how can we improve extruder to produce a plant-based meat with organoleptic parity to conventional animal products, or will it help make extrusion more high throughput? So that's the answer we're looking for from you. In the successful proposal, will include how you plan to improve extrusion processing or mechanistic analysis through a novel method. please be informed that the proposal focus only using traditional extrusion methods to analyze different plant proteins will not be in this scope, because we want to understand beyond the current existing state of the art, and we want to understand what else we can do to the machine itself to make it better. In the proposal, we also want to know which plants, protein, and ingredients will be evaluated, and how this will improve scalability, sensory, functional, and nutritional characteristic of the end product will be demonstrated. What is the biggest challenge facing right now in the scaling of your methodology that is predicted to be? Bonus point for any proposal that would help demonstrate the feasibility of extrusion processing innovation that you have in mind. Any pilot scale planning or any life cycle or techno-economical analysis of the processing methodology will be a plus point, because we want to promote and we want to support the technology that can scale and can be sustainable in the long run. So with that, I would like to pass the stage to my colleagues, Dr. Manasa.
All right, thank you, Asman. Good morning, everybody. And I'll be covering the funding priority B, which is developing tools and knowledge to promote stemness and proliferation in seafood cell cultures. Next slide, please. Can we have the next slide? Great. So just by way of some background information, I'd like to highlight where we currently are within the context of aquatic cell cultures for the purpose of creating seafood, cultivated seafood products. In comparison to where we are with mammalian and even avian cell lines, aquatic cell lines are at a slightly earlier stage. And this is sort of evident from the sorts of challenges we're facing in the lab with developing these lines. So, for example, doubling times of existing fish cell lines as well as other aquatic invertebrates are quite long. And there are other sorts of challenges with the lines not really showing desirable differentiation properties. They tend to either spontaneously differentiate in culture or not really differentiate down the myogenic or adipogenic lineage. But apart from the cell line challenges, we also are facing challenges with culture media, as is probably quite well known to a lot of you on the call. But apart from the cost issue, there is also the challenge of there still being a lack of not very well established serum-free culture conditions for a whole range of aquatic cell lines. And this sort of also goes hand in hand with the fact that our existing knowledge on the metabolic efficiency of the combination of the cell lines and media formulations is currently quite low. So if we just look at, next slide, please. So if we just look at where we want to be, that's essentially the opposite of everything I've just said. So we'd like to have efficient aquatic cell lines with doubling times that are more comparable to mammalian or avian counterparts. Cell lines where spontaneous differentiation is not a problem. And this is particularly pertinent as we scale up. And we would also like to see the availability of plenty of cell lines that can be easily differentiated down desired lineages of muscle and fat. And this goes hand in hand with desired developments in appropriate culture media formulations that are cheap and easy to access, as well as serum-free culture conditions that can actually support both proliferation and differentiation of a variety of these cell lines. And finally, this will underpin us obtaining a higher metabolic efficiency of existing cell lines in these novel media combinations to make sure that that combination is high and desired. Next, please. So really the purpose of this grant program within this funding priority is to build a sort of metaphorical bridge through which we want to use this grant program to bring together ideas, people, and funding to facilitate the journey from the left side of the screen to the right side of the screen. And if we just, next slide, please. So if we look at the people aspect of it, that would be all of you on call and your applications to the program. For the funding side of things, we are continuously grateful to our donors who have agreed to support this program year on year. And on the idea side of things, this is where we'd like to see not just this list here, this is by no means exhaustive, but we'd like to see approaches which tackle several of these problems. So for example, optimized basal media, cost-effective serum alternatives on the media side of things, or optimization of culture conditions, optimization of cell line properties, or even the generation of new cell lines with desirable properties that can help sort of address that challenge of inadequate cell line availability. And we also think that for several aquatic species, our fundamental knowledge of these species is not very robust to begin with compared to literature that's out there for mammalian or avian counterparts. So even aspects looking at the fundamental understanding of the metabolism of these animals, as well as better understanding of the cell types and tissue types found within them. And how we can even explore this concept of transdifferentiation and the generation of new cell types, new cell lines from cell types that haven't been explored in detail before. So those would all be buckets of ideas or concepts that would fit very well within the scope of the funding priority B. And I just want to highlight that underpinning all of this is a box that says research tools. And this is something that became, I think, evident to the industry over the last couple of years, which is that for several aquatic species, even fairly basic research tools such as antibodies or annotated gene sequences are lacking compared to mammalian counterparts. So we see this as something that has been quite understated in terms of the importance that it has in propelling our industry forward. So we'd like to really highlight this as a key priority for this funding bucket. So it kind of underpins any of the other approaches that you'd be looking to take for this priority B. Next slide, please. So what are we looking for in a proposal? Just to sort of home in on what I've just covered, one of the key questions that we'll be asking is, will your approach essentially help make fish or aquatic invertebrate cell culture less of a pain, really? And specifically, we'd like to see proposals which address multiple points rather than focusing on just one. The more sort of points that you can hit, the more robust and obviously the more strong it would be in helping the research community as well. So that would be looking at improving doubling times, metabolic efficiency, and cell line availability. On the media side, looking at reducing the cost, as well as the use of serum and animal-derived media components. And on the differentiation side, sort of underpinned by increasing our fundamental understanding of how the metabolism of these animals work and how the cell types function, preventing spontaneous differentiation. The second point that I want to stress a little bit more on is, what research tools do you need to be successful in order to answer these questions? And do they exist already? And if not, how do you plan to create them? So that could be an antibody, a primer probe, a sequence, an annotated gene sequence, etc. And if you do plan to create these new research tools, what are your plans to share them with the research community or share them with the wider community? So those are really the three main questions that we'd like to see in a proposal, and covering those three aspects would really make a strong case for your application. And finally, on the topic of species focus, the main species of focus for this year's priority B is fish or aquatic invertebrates. But that being said, we don't have like an exhaustive list of particular species. Any species really that is seafood relevant would be good to see. And bonus points if you're kind of able to justify why you've chosen the species you've chosen, whether that's because it's a popular seafood choice in a particular part of the world. Has it been overfished? Is it critically endangered? Is it a particularly high does it have particularly high carbon emissions in terms of the food processing? All of these aspects, if you're able to kind of take that into account in justifying your species, that would be really encouraged. And also, that being said, I want to highlight that we are also open to considering proposals that look at comparative approaches with mammalian or avian species as well. And what I mean by this is, for example, if you already have some data from, say, C2C12 cells or bovine cells that you've already got, and you seek to sort of compare how that data looks like in an aquatic cell line, by all means, your project can include that comparative approach. The second scenario could be, for example, if you have a novel idea that you've not tested at all before, but you would like to see how it performs in an aquatic cell line versus a terrestrial cell line, that would be encouraged as well. The only thing we want to make sure is that for this year, the main focus is not on just terrestrial species, but the main focus is on aquatic species instead, with considerations from mammalian or avian counterparts as sort of a secondary focus. Next, please. So if you'd like to learn more about these concepts and find a little bit more information about what our thinking has been like for these solutions, you can check out some of the entries we've got in our solutions database, which we've marked as primary and secondary here, and that's up on our website. And the second resource that you can look at is a workshop summary of a workshop that was held by our US team earlier this year. On precisely this topic, which is promoting stemness and proliferation in fish cell cultures. And this session was really informative in bringing to light the sort of severe lack of fundamental research tools for the aquatic cell culture field. So that really fed into us being able to expand the scope of this funding priority to not just focus on fish cell cultures, but also on aquatic invertebrates as well. So there's a lot of very useful information on there, and I would recommend you to take a look, and that'll help you gauge what approach you'd like to take, etc. So with that, I'd like to pass on to my colleague Dean to cover the next funding priority. Thank you.
Thanks, Mata, and good morning / afternoon, everyone. I'm a senior SciTech analyst for the APAC team, largely focusing on cultivated meat. So today I'm talking about funding priority C, which is looking at data collection and curation to inform the development of genome-scale metabolic models for optimization of feedstock formulation and feed conversion. And so as a disclaimer, I'll be discussing this priority in the context of cultivated meat research and its needs, but the principles of this priority and the applications you might seek to look at in your RFP proposals could be equally applicable to organisms that could be used for different fermentation properties. So keep your mind open if you're not looking at cultivated meat. This could still be relevant to you. Next slide, please. So one of the core challenges associated with cultivated meat, which I'm sure many of you are familiar with, is the cost of cell culture media. And so there's kind of two main approaches that companies or researchers can look at to improve efficiency here, which we have on the screen. So many people have been tackling this challenge through the lens of lowering the cost of the cell culture media. So that is optimizing for the lowest dollar cost per liter of media. Doing so focuses the attention on cheaper sourcing or cost of ingredients, such as switching from pharmaceutical or research grade to food grade ingredients, which are cheaper. So this is a very useful approach as it has cross-industry relevance. If you can figure out lower cost glucose, amino acids, growth factors, and other ingredients, then that can benefit everyone in the industry equally. However, longer term, we also need to think about this challenge through the lens of using less cell culture media ingredients in total. So this focus has a different metric, which is around optimizing the feed conversion ratio or the grams of ingredient required to convert to an equivalent amount of cell biomass. And so this research will take longer and be more specialized, as each of these formulations to optimize cell biomass accumulation will need to be tailored to a specific company's or researcher's cell line and the target product that they're making. And regardless of the challenge, this is still going to be necessary for the long-term success of the sector. Next slide, please. So the question therefore is, how do we tailor our media ingredients to the metabolic needs of cells? And we see this as critical to solving the challenge of competitively costed cultivated meat and fermentation-derived foods. Next slide, please. So to further highlight the importance of media ingredients to metabolic feed conversion's efficiency, we can look at this table, which is pulled from the techno-economic analysis that GFI commissioned a couple of years ago and recently published with our principal cultivated meat scientist, Elliot Schwartz, as a co-author. So this study looked at future media use scenarios for cultivated meat production at large scale. I think a facility producing 10 million metric tons of cultivated meat per year. So the study calculated media requirements for 3 different theoretical feed conversion efficiencies. And on the slide, we have our baseline scenario highlighted in red, and a low media use or more efficient media use on the left, and a less efficient use scenario on the right. And so that we can see that amino acids, salt, glucose, etc., these are all included in each of our media use scenarios. Next slide, please. And so the baseline scenario, we'll need around 46,000 grams of media to produce a kilo of cultivated meat. It's important to note, though, that most of this, I think around 45,000 grams, is water. So as a result, it was determined that a minimum of 600 to 800 grams of solids were needed as ingredients in media per kilogram of cultivated meat produced. So obviously, these numbers would need experimental validation, as these calculations were extrapolated from literature and company data that was submitted as part of this study. But we see this as a useful baseline scenario to work off of. So if we look at this calculation as a feed conversion ratio, we can see that this 600 to 800 grams for 1 kilogram of meat out would be about 3 times as efficient as chicken production and chicken feed conversion ratio. So this gives us initial confidence that this approach can have environmental efficiency savings that could be very significant compared to conventional meat production based on the amount of inputs we need to create kilos of meat out. So then if we look at our high culture media use scenario, which is on the right of the highlighted column. So this is a conservative calculation where through this publication, we determined that would see a carbon footprint increase of between 73 to 240% compared to our baseline media use scenario. So we can see that cells metabolism is not just important for bringing down media costs, but will likely also be very important from an environmental efficiency perspective of cultivated meat production as well. Next slide, please. So longer term, cost reduction is centered around optimizing the feed conversion ratio of cultivated meat production, which will rely on metabolic modeling. Next slide, please. So here we have a figure which provides a really good overview of the general steps or building blocks required to generate a fully realized metabolic model for an organism or cell type. So this framework can allow us to move to a more turnkey type system of analysis for metabolic model development. As a note, this slide was made by a GFI fellow, Sandra Gomez, who recently published a review paper on this topic, which is linked at the bottom of this presentation, and it's also in the concept note as part of the RFP document, which you can view. So if we look in the published literature, there's not a lot of info on the biomass composition of specific cells. So if you have a cell that weighs a certain amount, how is it actually divided into the different content of the cell itself? There's more omics data or annotated genomes available. However, every category in column 1 through 3 in the figure here are really critical in creating a genome scale metabolic model that we see in column 4. So a genome scale metabolic model, or GEM, is key in subsequently determining the media ingredients to optimize production, which are covered in steps 5 through 7 in this graphic. So these GEMs from column 4 are available for some species and cell types where you may want to, in an application, suggest data gathering for the downstream categories to make your metabolic model more robust, or begin to look at optimization strategies for media for a particular cell type. However, for other species, we may not even have the upstream metabolic models, and a project could therefore focus on this element of the data generation. So that then later, other projects could build off this foundational knowledge you generate to focus on columns 5 through 7 later. So in summary, the appropriate research focus for category C here is really going to depend on rather the species and cell type that you choose as to how you focus your proposal. So we know, for example, that zebrafish or Chinese hamster ovaries have really well validated gems available, but for many of the more commercially relevant species, this is not the case. And so it'll be important to focus on this in any proposal that you put together on this topic. And with that, I'll hand back over to Wasserman for our final topic.
Thank you, Dean. So for the final topic, we're going to look into improving feedstock availability for food fermentation in biomass and precision fermentation platform. As we know that the current fermentation platform is utilizing commercial setting is largely utilizing the source of carbon from biomanufacturing. Most of that are actually glucose, and those glucose are either coming from sugar beets, sugar cane, or maize. And this is very expensive extraction method to get those glucose, as well as it have a high potential risk for global warming contribution. And also utilizing those to extract glucose actually take away their opportunity to be used for any other food application for human consumption. So we know that microbes are robust and we can use microbes in many different ways. So microbes have the permissive ability to actually utilize different feedstock or different carbon sources. They have enzyme that is robust enough to actually utilize a repertoire of feedstock. However, for food, that can get a bit more complicated because the idea of using non-conventional feedstock is actually not new. However, with the food supply chain, we have to consider any contamination and any risk that will cause to the food system if we actually start looking into alternative feedstock. So therefore, for this particular topic, we want to look into microbe that we don't have the luxury of like using any feedstock with the food, because we might have the situation where when you look at agricultural side stream, this current setup of the agricultural side stream actually introduce other microbes to grow. And these microbes can actually produce toxin, or the condition that it grows create toxin within the feedstock itself. So therefore, when we actually move to use these feedstock to produce food for alternative proteins, those feedstock may not actually be safe for human consumption. So therefore, we are exploring the alternative feedstock for food production. We have to consider this situation and how whether these microbes will be suitable, and safety also has to be in consideration as well. So we are looking at the process to help with that. And so for us to explore that, we want to ask for a proposal to list out what type of microbe that you are interested in exploring, and what type of feedstock that those microbes can work together. We want to know why do you choose the pair of microbe and feedstock to explore to be a feedstock for alternative protein application. So we mentioned that these alternative feedstock could be polysaccharide, starch, so anything beyond glucose sugars, because we want to create a more sustainable food system, and therefore we want to explore any other potential that might be able to use for this. But in addition, not just the starchy feedstock that we want to look into, any microbe that can grow on 1 or 2 carbon compounds like waste alcohol, acetate, methane, carbon monoxide, or carbon dioxide that can eventually produce biomass, or can use the feedstock as to grow the microbe and create product for precision fermentation is within the scope of this topic. So in addition, we also look into the bioprocess of how you, like, of course, when you look at the microbe, you have to identify the feedstock that can feed that specific microbe. So we want to know why you choose the specific feedstock and what is the, microbe that would pair well with such feedstock. Additionally, with the microbe and feedstock, you also have to look at the bioprocess. The bioprocess decided can grow these microbes using this feedstock without introducing the toxin or the process that may create toxin. Or if your process have created some toxin, how would your bioprocess remove or eliminate that toxin? So eventually your product can be used for a food grade product or food grade or generally recognized as safe ingredients. So therefore, what we are looking for in particular for this topic is what fermentation microbe and feedstock are you interested in? Why are you interested in those microbes? You have to show us like what will be tested, what is going to be characterized, and which microbe will be developed. These microbes should have a potential for food safe fermentation or food safe fermentation derived products. And how those combination of microbe feedstock and your bioprocess design will actually create the food safe ingredient or protein biomass for the fermentation product. These microbes have to also answer to the price, sustainability, and availability over the current state of the art. Because if we start pursuing a very expensive feedstock, I don't think we're going to be creating a very sustainable food supply chain. We also want to see evidence at the metabolic level of why you were choosing a pair of specific microbe and feedstock. So we want you to be able to showcase those evidence. We also want to see your thinking on what do you think as a technical and biological challenges associated with the current industrial adoption of the bioprocess that you might be proposing. So we are looking forward to hear your thoughts and your proposal. And so right now, I'm going to move on to the step to apply for this grant. So all the proposal can be created and submitted through our application portal at grants.gfi.org. We will be sharing this slide later after the presentation. And within the proposal, you'll be asked to submit the following information. So your project details, your case of support, which can total up to 6 pages, comprising of 1 page for a track record explaining the qualification of the PIs and project team, and 5 pages describing proposed research and its context. The work plan of the proposal, justification of resource. So explain to us in 2 pages of how would you be using the resource. Your impact plan. So this means the 2 pages describing how the proposed will impact the project, will have include mechanism that project will have in place to maximize the reach and impact of your project. The resource may be requested to support impact activities, and the use of these can be described here. Any project partner letter of support is highly welcome. So up to 2 pages of any letter from partners who are committing to contribute financially or in kind of the resource for your project. The letter should outline how the partner proposed to contribute and the value of the contribution to the project. The assessment criteria of the proposal will be according to these 4 different categories. So including scientific excellence, applicant and partnerships, expected impact of the proposal, and your project planning. So we recognize that our requirement for proposal, it has to be written in English, meaning that many researchers may be reading in the second language. So this will be taken into account into our consideration when we are evaluating the proposal. We will not penalize researchers who may be writing in a second or third language. So we are actually promoting everyone to submit. This grant is actually open for everyone around the world to submit. And also, if you want to learn more of a specific topic and you want to talk to both the people on the webinar today, as well as our US scientist team, please sign up to the networking event. So the networking event for Singapore time will happen on the 25th of July at 9 a.m. So next Tuesday. So we hope to see everyone there. So as for right now, please check out FAQ link within the RFP document that you have already maybe downloaded. And if there are any other question, you can also address the question directly to the research grants at gfi.org, or you can ask your question now. Please make sure to use the Q&A box. And then we can, you can also upload the question in the Q&A that you want to be answered. And if it's going to be helpful, please help label what is the topic that your answer about. So with that, we can jump on into the FAQ section. So I have the first, yes, so the first question, the first question is if the presentation will be available. Yes, so we will be actually sending the presentation to the registrant, but I think maybe we can find a way to send it to all the register email. Yep. So I see the question regarding as a plant pulse processor, we want to explore value addition of pulses for plant protein from pulse and value addition of byproduct of pulse to feed nutrition. Please advise how we can proceed. So I would recommend that because within the scope of this presentation, we're actually looking into more the technological innovation. I think your contribution can come in terms of collaborators or with any researcher. So I would highly recommend for you to join our networking session to identify any potential collaborators. And I believe that this can either be in the extrusion part or even the feedstock part, because I understand that you probably have a lot of side stream that you can explore as well. So I hope that's answered your question. I would highly recommend for you to go to the networking session to identify your different potential collaborators that you can work with towards this RFP. The next question we have, would the competitive grants process consider proposal for developing plant exclusive feeding strategy for dogs? I believe that would not be within our scope, unfortunately. Yeah, so we are looking mainly for human consumption, and we would like to replace the human meat consumption. So the feeding dogs and cats would not be within the scope. Does partnership between two industry players, non-academic, count as well for RFP? So yes, so anyone in either academics, industry, startup, postdoc can apply for this RFP. So for your partnership to actually help activate or unlock the additional 100K grant, you have to be able to showcase that first, your partner have a significant contribution to your work, as well as the fact that the partner is not yet within the alternative protein space. So the next question is, would the grant consider any other process, not fermentation, that is using leaf biomass feedstock to make protein and fiber? I believe this would not be within the scope of these topics. So unfortunately, I think that might not be, because if you can explain a little bit more of what your process is like, but so far from your question, I don't believe that would be within the topic of the topic D, unfortunately. So feel free to add any other questions if you have right now. We're gonna give it a few more minutes, but make sure if you want to talk like one-on-one to either myself, Dr. Manasa or Dr. Dean, as well as some of our US scientists, join us at the networking events. And you actually can have like a one-on-one session for about like 8 minutes for each scientist. To actually have a conversation and maybe scoping out your ideas for this proposal.
I just wanted to add a quick point for the question that says, does partnership between two industry players count as well for the RFP? So just to sort of delineate that better, for the extra 100,000 that you can request for, that applies to if the collaborator, as Wasimon mentioned, hasn't worked on alternative proteins before, and if they're a new player. But that being said, you can also submit an RFP for two alt protein industry players that are already established. That's fine. It's just that you won't be able to claim the 100,000 extra, but you can still submit a proposal and they can count as your collaborator as long as you're able to kind of clearly justify what impact each collaborator is going to bring to the project. So just wanted to make sure that's clear. Like, you don't have to have a collaborator who's new to the field to apply. It's just that for the extra 100,000, that is the prerequisite.
I believe we don't have any other.
Oh, there is a last question.
There's one question. Can one proposal each submitted for different categories under the same PI? So I think that you can submit. So the process of the proposal, we will have our the expert under each topic review whether your proposal is within the scope. So for sure you can submit the proposal. However, we will evaluate whether each of the proposal you submitted is within the scope or not before even move on to the actual review process.
Yeah, and that they are significantly also unique from each other. If it's the same thing that's submitted through different lab members, that would not be considered.
Yeah. So I mean, even when you actually download the full article of our RFP, you would know we actually listed quite clearly this year of what is not within the scope. So if you're gonna plan to submit two proposals to address two different topics, you have to be able to answer those specific questions under different topics completely for your proposal to be considered, I think. Yep.
I think maybe it's also looking at if one proposal could be applied to two topics. So say B and C if they're using a seafood cell line. So I think maybe in that instance, you could address both B and C in the one single application, and that would strengthen it considerably in that you're hitting even more of our required topics for the same amount of money. So instead of splitting it to two applications that are the same but addressing different topics, have one application that you can show how it addresses both topics, and that would make it more competitive.
Yep.
And there's one more about, are there any limits on the number of proposals? I don't believe so.
I don't believe so. But we want strong proposals. So like if your proposal is not strong enough, it might be like it might not go through the first stage of the review. I believe we are good for today. I don't see any other questions. So thank you everyone for joining us today. So make sure to sign up for a networking event if you want to discuss more in detail. Any other questions you may have, or it might come up after the webinar today. We'll look forward to see you there. Thank you everyone for joining.
Thank you.
Thanks everyone.